Thanks for the extremely kind introduction. So I'm going to talk about mutation rates. And this is a work done by my student, Gabriela Lubinska, who is here. Let's start. So here's a question, very basic question. You know, there's no evolution without mutation. And the question is, what should be the rate of mutation when an organism replicates? And a very early and classical observation is that if you look at many genomes, many organisms that vary in their genome size and vary in the mutation rate, they each vary over a whole range of several logs, but the ratio between them varies less, so that you get on average a typical number of mistakes per genome replication. And that, of course, has the implicit notion that the mutation rate, if it varies across organisms, then at least within an organism, it would be a characteristic number, a constant of that organism. But it doesn't have to be the case. It could be that mutation rate would vary, and it could vary. It could vary, for example, across ecological conditions. This is a very well-known phenomenon that Lilach and others have studied extensively. It's called stress-induced mutagenesis. The very simple notion, when times are bad for the organism under stressful conditions, etc., it would do more mutations. That's very natural. What I want to explore today with you are two additional deviations from a constant mutation rate. One is that the main one that I'll talk about today is that it is possible, and there are indications that mutation rate could differ between individuals in the population. So not only between the two of us, that would be quite obvious because we are different genetically, but perhaps also among individuals that are perfectly genetically identical to one another. Are they still exhibiting variation in their mutation rate, and what could be the functional and evolutionary implications of having a diversity of mutation rates explored within a population of even genetically identical individuals. And the other dimension is that evolutionary rate could be different along the coordinates of the genome. So if we say that mutation rate in human is 10 to the minus 9, that's the probability per base per replication to have a mistake, maybe that probability is different for different genes. Maybe some genes are predominantly, originally, are more prone to make mutations than others. That could bias the search towards the genes from which the organism might need a solution right now. So I'll devote most of the talk to variation within a population, but then I'll show you experimental work that aims to test whether different genes in the genome exhibit different mutation rate, and are they biased in any particular way to give the organism what it needs at a certain moment. So first you can ask if cells are genetically identical, and we're mainly going to talk about microbes, how could they manifest any variability that is not encoded in their genome? Or do they, in fact, manifest any variability? And the classical result along these lines is that of Elowitz. He looks here at E. coli bacteria. Each cell is a little dot here, and the cells are expressing a green fluorescent protein, and although genetically identical, you see that different cells express the same protein to different levels. That's a very, very interesting observation. We and others, more than a decade ago, have expanded upon that. This is a foreign gene transplanted in the genome of E. coli. We asked what happens to the natural genes of the genome. In this case, in the yeast, it's a eukaryote as opposed to E. coli. So we looked at many genes under many conditions. This was done with Naama Barkai, with our share student Alan Bar-Even. We looked at many genes in many conditions, and for each gene in each of the conditions, and the conditions could be high temperature, low temperature, antibiotics, etc., we asked what is the mean expression level of that gene across many genetically identical cells, and what is the noise, the coefficient of variation among the cells in the population. If all the cells express that to the same level, the noise would be zero. And what we see is that there is noise, and the noise scales interestingly with the mean. There is a precise statistical reason for that, which you understand. But interestingly, many genes are either above or below this scaling line. which means that for some gene, it appears that biology selected for a deliberately high level of variation, given the mean and the general scaling. And for other genes, evolution appears to have selected for diminished variation, so that the entire population behaves very similarly with respect to some genes and very differently with respect to other genes. And when we looked at the nature of the genes that are above the line, it turned out that many of them are involved in stress response. When the environment is unpredictable and stressful, perhaps the best thing that you can do is to generate variability among your otherwise genetically identical cells, so that although they have exactly the same genome, they would each gamble on a different level of expression of the protein, and then you would get some selection based on phenotypic rather than genetic variation. Okay? In fact, earlier than our observation, Nino suggested, based on totally theoretical considerations, that cells might be, even if genetically identical, they might be different from one another in each particular protein, not just by amounts, but by the sequence of the protein too. How could they be different in the sequence of the protein if the sequence of the DNA is exactly the same? Well, all the processes, including transcription and translation into proteins, are very stochastic processes. The error rate in DNA replication is orders of magnitude lower than in transcription and translation. Mistakes happen when the ribosome reads the code. And in fact, what Nino suggested is that a particular set of proteins, those that are responsible in their own term, in their own for replication of the DNA, they might be subject to deliberate translation mistakes in their own production, so that although identical cells in population have the same gene that would replicate the DNA, it gives rise to slight variation in the protein sequence, such that perhaps some of them replicate the DNA with a higher propensity to make mistakes, and others replicate the DNA with a lower propensity to make genetic mistakes. So this is a propagation of mistakes from translation, from variation in the phenotype. If it happens in the protein that replicates DNA, that would give rise to a variation in the rate at which genetic mutations are seeded in the genome. And this was a theoretical prediction. In fact, the prediction was that the so-called mutators are strains that have a higher probability to make mutations in the DNA. So far, until that paper, mutators were thought of as a genetic attribute due to a genetic mutation in the protein that replicates DNA. That particular strain copies DNA with a higher error rate. So the idea was that transient mutators that have their DNA sequence of the DNA copying machinery perfectly okay and perfectly like the others, transient mutators are those that have acquired sufficient amount of translation mistakes in their own DNA replication machineries, such that they would give rise to higher or lower rates of genetic mutations. So the Nino prediction is that most of the mutators that we see in populations are actually transient. They do not result from genetic mutations in the DNA copying machinery, but rather in phenotypic mutations in the DNA copying machinery. Very recently, we have done, and I don't have time to go into any of the details, we have done the first survey that allows to actually measure what is the rate of translation mistake in each of the genes in the genome. Okay, so each dot here is a gene in the E. coli genome. The x-axis is how highly or lowly that protein is expressed in the cell, how many copies per cell. The highest is millions of copies, the lowest is less than 1 copy on average per cell. But the interesting thing is the y-axis. The y-axis is how often does that protein get a mistake upon its own translation. And you see, again, a general negative correlation. The more highly expressed the gene is, the more it is protected from being translated with mistakes. That's an indication that mistakes in translation are themselves somehow programmed into the sequence of the gene. But what's very interesting is that if you look at DNA repair protein, those that have to repair mutations that were seeded by the DNA replication machinery, they have a particularly high level of translation mistakes, especially given the general scaling between those two variables. So somehow it seems that biology wants to diversify its genes, its proteins, sorry, even if the genes are the same, not only with respect to amounts, but with respect to the actual protein sequence that they have amidst a constancy of the genetic material, the genetic sequence. So with that, we turned into asking whether indeed there could be a variation either due to amount or due to sequence in the tendency of isogenic cells that are genetically identical, to have different mutation rates. So what could govern the mutation rate of an organism, of a particular cell? Again, those DNA repair enzymes. Those enzymes, their role is to repair mistakes that happen in DNA replication. They do that immediately after replication, before the wrong version of the DNA is inherited to the daughter cell. So they have a very narrow window of opportunity to fix mutations. And as you can imagine, the higher the level of those proteins, the lower is the actual mutation rate that is inherited, and vice versa. Okay? So a recent study, not so recent, but a study I'm forgetting his name, from Harvard. I should be able to get that. Do you remember the last author's name? It will come in a few minutes. He actually asked that question. How variable is the expression level? He didn't look at the sequence as we do now, protein sequence. How variable is the expression level of a key protein in the E. coli genome whose role is to repair mistakes in DNA? Okay, so here is a bacterial cell, here is that protein, and it could be present in 1 copy per cell or more than that. The interesting thing is that if you look at individual cells in the population and you go to each of them and you ask how much you have from that protein, the protein is called ADA, it turns out that there is variability. Considerable variability. Part of it is because of a positive feedback loop that this protein exerts on itself. The bottom line is that different cells have a different level of expression of that protein, although genetically identical in any respect. And they have actually measured and saw that the variation in expression of that protein indeed translates, negatively correlated in fact, with the mutation rate that each cell exhibits. So if you have a lot of this protein, that cell would typically replicate the DNA with a lower error rate than others. So, but the question that Gabriela and I, and by the way, we know a lot about the statistics of such variations that are not genetically inherited in the genome, but rather result from stochastic effects that could be amplified or repressed by genes, as I've shown you from our previous investigations. So there could be several regimes that could be thought of. Perhaps these are 3 different cells, genetically identical, and the first happened to be, at the beginning of a time course, happened to express a protein at a high level, someone else at a low level, etc. There could be a situation of so-called no mixing, such that each cell would keep its identity, high or low or medium level of expression of that protein, despite being genetically identical to all the others. And the mixing time could be very short, alternatively. For example, the shortest that it could be is something in the order of the cell doubling. Once the cell is replicated, perhaps there wouldn't be a lot of memory, or some memory at least, of the variation in expression that it's inherited from its parents. So different cells have different mixing times. And as we saw, different cells, this is in general, and Uri alone has shown that this is indeed prevailing in many particular genes that we look at. And we know a lot about the statistics of cell-to-cell variation in amounts of proteins, and we know a lot about the statistics of the extent of memory of such variation in amounts. Okay? So here is all the facts that you should know in the background. There is a gene in E. coli, it's called ADA. It's responsible for mutation rate by fixing mutations. Different cells have different amount of that protein. And the daughters of a cell that has a high or low amount of the protein would have some vague memory of their parents' amounts because of simple inheritance of the content of the cytoplasm, but there would be some mixing of that information such that over time they would ultimately forget, after a few generations, they would forget what was their parents' mutation rate. So the question that we wanted to ask, and at the moment it's very theoretical, although we are aiming to construct an experimental paradigm that would test that, what does it do to an evolutionary process if you have or you don't have variation in mutation rate among genetically identical cells in the population? That's the question. And we are playing a game. It's a very known game in theoretical evolutionary biology. There is a fitness landscape. It's a function whose domain is the genotypes, and there's a function of fitness ascribed to each. The fitness is the doubling time, essentially, of the organism. And we are playing a mutation selection process. The mutation means that you change your genotype, and selection is according to the resulting phenotype. So we do that both with simulations. There is a theoretical embodiment of that. We do all sorts of Gillespie algorithms, etc., etc. I show you some results that are obtained with simple-minded computer simulations in which, for G generations, we calculate the fitness of each individual based on its genome. We ascribe to it a doubling time, and then there is also mutations. And the populations are different in the sense that they each have a different schedule of inheriting variation in mutation rates. And we play with fitness landscapes that can be very smooth and very rugged. Rugged means that you have lots of local optima, and smooth means that you have typically very few. And the whole study is actually focused on 4 alternative regimes of inheritance of mutation rate. So the simplest regime is fixed mutation rate. Everyone has the same mutation rate because they are genetically identical. What else you would expect? The second possibility is that there is variation in mutation rate in the population. There is a histogram of mutation rates, presumably a histogram of the expression level of the gene that determines mutation rates. But each cell samples from that distribution, and there's no memory. So the next cellular generation samples with no memory of the parents from the same distribution. Another possibility is that there is variation, but it's genetically encoded, perfectly genetically encoded, so that if you were a mutator, your offsprings would be mutators unless they happen to have a mutation in the DNA that changed that property genetically. And lastly is what we're most interested in, and that's the new notion, is that there is inheritance in the mutation rate, but it's imperfect. And it's exactly that imperfection of splitting the content of the cytoplasm that would determine that if you had a particular mutation rate sampled from the distribution, your offsprings would have some memory of that. They would choose from around that place in the histogram, but it wouldn't be necessarily exactly the same value because the content of the cytoplasm, there's nothing that ensures that it's split equally between the 2 daughter cells, but it wouldn't be memoryless either because nevertheless, after all, if you inherited from a rich parent, you're likely to be richer, at least if you are the immediate next generation. Yeah.
So, just from this perspective, should I think of like mutation rate as a little bit, I think of the same machinery as like the learning speed, and the learning you typically want to reduce step size as you get closer and better and better into fitness or objectives. So is that basically a mechanism that tries to replicate that?
That's exactly the point. In physics or in computer science, in fact, too, this is called simulated annealing, right? You reduce your tolerance to mistakes. So a teaser. What happens, what I'll show you, is that this happens, although it happens naturally, without us instructing the cells to do that. But the teaser to the teaser is that something more interesting and profound than simulated annealing appears to happen here. Okay, but this is exactly where this is going. Okay, so how do we experiment computationally with that? As I said, we are actually hoping to generate in the lab versions of E. coli that would realize each of those, realize each of those regimes. But we don't have that yet. So I'm going to show you computational results. And in order to do that computationally, we have to devise computational genomes and ascribe to them fitness. And we want to ascribe to them fitness controlling the ruggedness of the fitness landscapes. Luckily, there are mathematical procedures to do that. For those who know, these are called, for example, one of them is called the NK model by Stuart Kaufman. And the NK model is a way to, the K in the NK model, the N specifies the length of the genome, but the K specifies the ruggedness of the fitness landscape. So when K is very, very low, we are talking about fitness landscapes that look like that. And when the K is very high, the highest that it can be is the length of the genome, like N, it would be very rugged. So we are experimenting, so we let organisms solve a fitness landscape, and it could be either a simple one or a more rugged one. We place them either randomly or in a fixed position in this landscape, and then we play the process of mutating them, mutation selection. So they are allowed to mutate, typically to the individual neighbors, but according to the mutation rate. And they are replicated according to how good they are. That's very simple. Okay? So if we are plotting as a function of the generation time the mean fitness behavior of a population, then we typically see an improvement that's obvious for every evolutionary process. But the question is whether the 4 different regimes of inheritance of mutation rate do any differently in the different landscapes. Okay? And of course, we do many repeats in any of them so that we gather statistics, because this is a very stochastic process. And the first result that we got was that indeed, if you look at this landscape, the the noisily inherited mutation rate, that which we think fulfills the best the observation from the experiment, namely that there is variation in the population, but there is some limited memory, not zero memory, not perfect memory. There is some memory of the mutation rate of the parents. These appear to perform the best. This is the red colors. I hope you see them. Appear to perform the best in solving this fitness landscape. If you have a fixed mutation rate, it doesn't work so well. If you have perfectly inherited mutation rate, it gives you some benefit over the fixed mutation rate. And if you have randomly inherited mutation rate in each generation with variability, it does a little better. But at least in this particular setup, the noisily inherited mutation rate, again, this regime that we think fulfills the best what we see in observation in the cells, appears to be doing the best. So is it an indication that this is the reason for why this mutation rate scheme was inherited? Of course, it was selected for in evolution. Of course not, but it's some indication that it can do something useful. Yes?
So is it the case that you kind of tune all these 4 cases, the hyperparameters, so you say you fix the mutation rate, you choose the best rate to improve fitness with each one of those, you choose the best? How are you comparing the different?
Okay, so nothing was tuned here, and in particular, if you asked whether we enforced them to do something like adaptive learning or simulated annealing, etc., we haven't forced them to do anything. I'll show you in a few minutes that they appear to exhibit, but this is something that we didn't instruct them to do. They appear to exhibit something that looks like simulated annealing, and actually beyond that, as I'll say, but we haven't instructed them to do anything. Everything that we told them, I told you so far. There are 4 different regimes of inheritance of mutation rate. It could be constant, not constant, variable with different levels, etc., etc. And we simply run the experiment many, many times, the experiment of mutation and selection. Okay?
Can the strategy itself evolve? So you start from fixed mutation rate and allow the
That's
system to evolve?
interesting. That would be interesting, but we haven't tried it. Well, if you move into more rugged landscapes, it doesn't always behave as clearly, and there is more variation in the performance. This is the most rugged landscape. But even in the most rugged landscapes, it seems that the noisily inherited mutation rate, variable mutation rate, appears to be doing better than other strategies, either in the sense of the fitness in the final at the steady state, or the rapidity at which they reach that maximal fitness. So we wanted to understand indeed, why does it do better? What happens there? We haven't instructed them to do simulated annealing, adaptive learning, or anything like that. Everything that we told them that is at their disposal, I already told you. So we wanted to understand, what do they actually do? So luckily, since this is a simulation, we can record whatever happened in the process, and in particular, we can ask for each run of the simulation, each run is an individual curve here, what was the actual mutation rate that was exhibited in the population at each time point? And we looked to compare this noisily inherited mutation rate into perfectly inherited mutation rate. And what appears in both of them is that when the simulation begins, they typically start with a low fitness, they increase their mutation rates. But gradually, after some point, especially the red population of the noisily inherited mutation rate, they start to decrease their mutation rate. Although we haven't instructed them to do that, they appear to be doing something like simulated annealing, in which you cool the temperature, you cool your system, so effectively this is comparable to lowering the mutation rate. But the interesting thing is that whereas the perfectly inherited mutation rate is essentially stuck with the high mutation, the highest that it could get, and it cannot explore further, and it cannot reduce further its mutation rate, the noisily inherited mutation rate appears to select for reduction in mutation rate more or less at the same time where the fitness becomes better. So we realized that we can, Gabriela realized that this process can be partitioned into two phases, the adaptation phase and the fixation stage. This is very similar to the exploration, exploitation, and the multi-arm banded, I think you know about it, scheme in which, you turn between exploration and exploitation phases. It appears that the noisily inherited scheme, again, the only rule that it has is variation, and the way that noise is being inherited in biological systems appears to be doing that and transition from the adaptive part to the fixation part. Most naturally, the perfectly inherited mutation rate is simply stuck with a high mutation rate once it finds a solution, and that actually hampers the performance of the algorithm. This has reminded me of something that I think many of you, especially the Israelis in the audience, know. This is the beautiful song by Pugi. What happened when I first time climbed the Everest mountain? I simply returned back and went backwards. So this is what happens to the perfectly inherited mutation rate. You have a good mutation rate, a high mutation rate. It allows you to climb, but once you reach the top, you keep going with the same speed, and you cannot utilize or exploit the fact that you have a good solution. So somehow I think that Pugi thought about something along these lines. Can I humbly suggest some implication to algorithms? So as you said, indeed, this is very reminiscent of simulated annealing. In simulated annealing, you're trying to optimize a search process. Typically, you try to minimize energy. Of course, here we try to maximize fitness, but it's essentially the same thing. In both processes, the danger is that you would be stuck in a local optimum. And in order to avoid local optima, what you do in simulated annealing is that you have a tolerance, temperature as a metaphor, that declines with time, so that at the beginning of the process you explore more, and at the end of the process you exploit on the exploration that you did. So it seems that our noisily inherited mutation rate scheme that is observed, that is again, I think, a natural reflection of what is observed in nature, but it's obtained very, very, very cheaply without any mechanism that instructs it, appears to be doing something like simulated annealing, because you increase mutation rate, increase the temperature, and from a certain point on, there is selection for reduction in mutation rate. Indeed, as I told you, at the various landscapes of the various ruggedness, from the lowest ruggedness to the highest ruggedness, we see an increase and then a decrease. If there is a perfectly inherited mutation rate, there is an increase in mutation rate with apparently any lack of any ability to reduce the mutation rate. So they are doing the Pugi thing. They climb to the Everest, and then they continue with the same speed and escape the top. So we wanted to see, though, do the cells do something more interesting, perhaps more profound than simple simulated annealing, and maybe this could teach us something about simulated annealing as well. So we did the following computational experiment. We ran the same landscape twice. In the first time, the cells are allowed to do what they are doing, and I told you. So they do the red behavior. They solve and they do simulated annealing. They reduce the mutation rate, etc., etc. Then we do the same run again, the same landscape. We ask them to solve the same landscape, same initial conditions. But this time, we do not let them choose their own mutation rate. We take the schedule of reduction of mutation rate from the previous run, and we impose it on the new population, the new evolving population. Okay? So they have simulated annealing, because what the previous population did was something like simulated annealing. They reduced their mutation rate. So they can reduce the mutation rate. They should be doing better than a fixed mutation rate. Indeed, they do better than a fixed mutation rate, but they don't do as well as the natural population. This is in purple here. They don't do as well as the original population. So it seems that the original population does something better than simply reducing the mutation rate with time. It seems to be doing something that I think, injects metaphorically high mutation rates when needed and low mutation rates when needed with respect to their place on the fitness landscape, so that it suggests that perhaps a naive scheme of determining ahead of time what would be the schedule of reduction in mutation rate is not the best that you can do. You can do better if there is free for variation in mutation rate and soft inheritance of that mutation rate, as opposed to strictly fixing a constant mutation rate. And there's more comparisons that we did along these lines, but I want to summarize this and very briefly move into a very short part, as I promised. So what we're suggesting is that noisily inherited mutation rate, this NIMR thing, could be a newly recognized strategy of variation in mutation rate in population. It has certain advantages, and it resembles simulated annealing, but it appears to do something more profound than that. And the comparison to stress-induced mutagenesis, and I think Lilach's immediate response when I described it first to her was that this is indeed similar or should be discussed in comparison to stress-induced mutagenesis. Nevertheless, I think it's different, and it's interesting to understand in what ways is it different. First of all, the mechanism is totally different. If you care about what the cell does in each case, then in stress-induced mutagenesis, it must be able to sense the environment, sense its own fitness, translate its own adaptation to that environment into a strategy of use of mutation rate. Typically, cells have different machines that copy the DNA, one that is very accurate, one that is very sloppy, and et cetera, et cetera. We have 9 versions of them in our genome. Bacteria have less, but nevertheless, they can vary the mutation rate, and there has to be sensing and a decision which replication machinery with what accuracy should be activated, such that when things are bad, the sloppy machinery would be used and vice versa. So this is one difference. The difference is in the mechanism, because in our case, there's no mechanism. There's no sensing. There's no, there's no different machineries with different accuracies that need to be invoked with respect to, in response to the environment. It happens naturally. And on top of that, I would dare to say that perhaps in stress-induced mutagenesis, as long as things are good, mutation rate is low, and when things are bad, mutation rates are high, whereas in this noisily inherited mutation rate, perhaps something more gradual happens. So towards the very end, I want to show you very, very briefly something that I think complements nicely the picture, and this is, if I've shown you so far, so this is stress-induced mutagenesis, this is what I've just shown to you. What about difference along the genes in the genome? So far we looked at the entire genome as one entity. And if you increase mutation rate, you increase it both for the genes for which you want to find new mutations, but at the same time you increase the mutation rate for the genes that are working perfectly for you. So it's not obvious that any increase would be beneficial. So think about it, how cool would it be, or hot would it be, if the cell had a way to increase the mutation rate just for a few genes and keep the rest the same? How would it know which genes to increase mutation rates for? It doesn't have any wisdom. It's the same machinery that copies everything. And the machinery has a fixed, within a cell, within a condition, a fixed mutation rate. So imagine, for instance, that cells had that extra wisdom of telling themselves, I'm feeling hot now, I'll express to the RNA level the genes that are needed to cope with high temperature. And if there was a way to insert mutations into those genes while keeping the rest of the genes cold, metaphorically and not metaphorically, that would have been an interesting trade-off between variation where needed and fixation or conservatism where needed. But how would cells do that? The DNA replication machinery is the same, at least when it runs along the DNA. So the thought was that perhaps something interesting can be there. Cells don't appear to have a means to go into the genome and rewrite it according to the information about the sequence of the gene and the environment. But what they do have is a mechanism to go and read the DNA differently. This is called transcription. The process of converting DNA information into RNA information is, of course, very gene-specific and very environment-specific. So only when the temperature is high would you go to the heat shock response genes and transcribe them into RNA. Now, RNA, without going into any chemistry, is a very interesting entity because it responds to the environment, unlike the DNA, which is by and large not responsive to the environment. It responds both by amounts. The amount of RNA is copied from a gene, for a Hitchcock gene under Hitchcock environment, is different than for that gene under other conditions and for the other genes in that condition. And RNA also goes a lot of interesting chemistry, and it's mutated while being generated. As I showed you before, not only translation mutation rates are very high, transcription mutation rates are very high too. So RNA is a very effective means to explore sequence space of the genes that are needed at a particular environment at that particular environment. There's a lot of variation that could be subject to evolution, but the problem is that RNA are degraded after a few minutes, and all that variation, including all its potential benefits, is gone. It turns out, though, that there is a process called reverse transcription that allows to write back into the genome some of the RNA molecules that were generated in cells. Typically, RNA viruses have taught us originally, historically, about that process, but it turns out that cells have those machineries either because of old viruses that landed in their genome or for other reasons, and they use them especially to copy those retro elements. And once you have a cDNA, a complementary DNA generated from an RNA, it can do one of two things. It could either overwrite the original version of that gene, like in a computer memory that you overwrite a variable, or it can go into a new location in the genome, into a new designated writing area in the genome, and put that piece of DNA as an additional copy, perhaps reflecting a mount variation in the RNA into a mount variation in the DNA in the next generation. So our beloved yeast cells, and I'll show you one more result, and with that I'll finish. Our beloved yeast cells appear to have such a machinery. That machinery is called the TY retro element. It's like a virus, essentially, and it builds itself, but it's not a virus. It builds itself a viral-like particle, this hexagonal, I think, or whatever it is here in the cell, it's not the real shape, in which this element takes care of its own replication. How does it replicate itself? There is DNA, it goes into RNA, it encodes for its own enzyme that converts RNA back into the DNA, and it has an enzyme that inserts back this DNA either at the expense of the original copy or in a new location in the genome. And this is how it propagates. We also have it. We have 1 million copies in our cells of this machinery, a related machinery, not this particular one, in the human genome. So we asked a very simple question. This is a parasite on the cell. Do cells parasitize on their own parasite in the sense that they would take their own RNAs, not this element's RNA, would hitchhike the machinery by integrating them into this viral-like particle, convince the reverse transcription enzyme to reverse transcribe them as well, and perhaps also convince the integration machinery to put them back into the genome. If they do that, then they could enjoy a higher mutation rate exactly on the genes that are transcribed at a particular moment, keeping the rest of the genome relatively cold. Okay, this is work in progress, but we have done quite some work towards deciphering that. It involves a lot of biochemistry that you don't want to hear about, but we taught ourselves how to pull out those viral-like particles from the cell and sequence both their RNA content before it's being converted into DNA and their DNA content. Turns out that some genes of the genome indeed undergo this process. They can get into the VLP at the virus particle at the RNA level and be converted into DNA. And it's yet to be shown whether indeed that DNA is integrated back into the genome, because otherwise it's meaningless, so that indeed they would manifest variation in mutation rate, especially in the genes that are needed at a particular environment, because only they have an RNA in the cell. So with that, I'll conclude and just thank Gabriela for doing all the first part that I've shown you, and the other people in the lab, and thank you, and I'm sorry if I've taken too much time. Thanks.